If you have a really cool new probe, say from Inspec or IMEC, that is designed currently for a bioreactor, that's one probe per bioreactor. But if you want a 96 well plate, you need 96 of them, so it also has to be cost effective. So you don't want the cost of your well plate now, if you have sensors, to be $10,000. So that's another challenge, the miniaturization and finding a cost effective solution. I think that's probably the biggest limitation that we're seeing at the moment.
Welcome back to the Smart Biotech Scientist. I'm your host, David Brulman, and this is part 2 of our inspiring conversation with Tom Valentin about small-scale perfusion cell culture. Tom is a group leader at CSEM for automated sample handling within the Life Sciences and Industry 4.0 business unit. In our first segment, Tom walked us through the revolutionary approach to miniaturizing perfusion systems in 96 deep well plates. Now we're taking it even further, diving into the world of process analytical technology for continuous bioprocessing. If you've ever wondered how to extract meaningful data from weeks of perfusion culture, or how to implement PAT strategies without breaking the bank, you are about to get answers that could revolutionize your approach. Tom's unique position at CSEM has given him insights at the intersection of academic innovation and industrial application. Insights you won't hear anywhere else. Ready to transform your perfusion strategy? Let's jump right in. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brulman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech.
Out of curiosity, I'm just thinking about the PAT capabilities and, looking at these parameters in real time. Since you have very small volumes in your deep well plate, and I would say since it's agitated very well, probably we can assume it's homogeneous. To what extent could we use optical probes or sensors integrated in the lid to, for instance, measure cell density? Where's the technology today?
Using optical density, like in the Mobius Breeze or the CSEM, that could also be used. So for the Mobius Breeze, it's in a microfluidic chip. So that's simple. It's a flat surface. It's flowing over it. In the CSEM, they have a 24 deep well plate with, as far as I can tell, a flat bottom that allows you to image through the bottom. Most of the applications we've seen use kind of pyramidal or round bottom well plates, which makes imaging much more difficult. And they're also made out of plastic, which is not ideal for imaging through. If we were to integrate it into the lid, we would then need to pull a sample out, image it, and then it's kind of like sampling. You could do that with a liquid handler. I think what would be cool is to get an optical probe that sticks down into the well. Now, whether you use optical or if you can develop a much smaller capacitance sensor depends on what the needs and what the preference of the user is. I think if you can develop a small enough capacitance probe that fits inside, that seems to be the gold standard, and I think that would probably be the most accepted by the industry. What are your thoughts?
Well, capacitance definitely is accepted, and I would say it's fairly easy to use. And probably, as you just mentioned, it could be a good compromise, especially at that small scale, because I think there's always a trade-off. Obviously, you want to have a precise method, but at small scale, you have certain limitations. But the upside at the small scale is that you get to test a lot of different conditions. So perhaps there is somewhere an in-between space. You get some real time. It's much better than what we have today, and it's sufficiently precise.
And the other challenge is when you're shaking a deep well plate, the liquid is not sitting at the bottom. So you don't actually have that deep of a sample. A lot of it is being pushed to the side. And so you have to position the sensor actually in a much smaller volume at the bottom of the well plate, or it's then maybe close to the side where the liquid will flow across it once every rotation. But that seems to me to be less reliable. And so the shaking helps with the agitation, keeping DO and keeping everything homogeneous, but it also poses extra challenges because the depth of liquid that you have to work with. And same for pulling your harvest and the bleeding. You have less space to work with that in these deep well plates because it's being spun to the side.
Yeah, that's a good point. There is definitely optimization needed, or perhaps you need to slow down the agitation for a quick moment to draw a more representative sample. Yeah, I agree with you. There is a limitation. Now, as we zoom out a bit, if you look at PAT in general, whether it's a small scale or a very small scale system in a deep well plate or a bit a bigger one in amber and other automatized bioreactors, what do you see as the biggest challenges in implementing PAT strategies and then to use also the data we're generating in a meaningful way?
I think, again, it comes down to the miniaturization, getting the technologies that exist to be smaller. And if you have a really cool new probe, say from Inspec or IMEC, that is designed currently for a bioreactor, that's one probe per bioreactor. But if you want a 96 well plate, you need 96 of them. So it also has to be cost effective. So you don't want the cost of your well plate now, if you have sensors, to be $10,000. So that's another challenge, the miniaturization and finding a cost effective solution. I think that's probably the biggest limitation that we're seeing at the moment.
Yeah, cost definitely is a big one because especially if you want to test dozens of deep well plates and if you have to pay $10,000 for each plate, I'm not so sure if you're going to run that many tests.
Exactly. And unless the sensor is reusable, but I don't know how compatible these are with being autoclaved. I haven't necessarily asked these companies, but if it is, great, then maybe you can clean it and reuse it. I don't know, from an industry perspective, is a single-use sensor more attractive or a reusable one for this type of deep well plate format? Because you said you did a lot of experiments with these 96 deep well plates for fermentation. If you had this, would you prefer a single-use or reusable?
I would say from an operational point, single use definitely would be much easier. You don't have to worry about cleaning, sterilization, and so on. But yes, I think we have to be very mindful about the cost. Perhaps single use would be too expensive. I don't know. But I think if I could choose my preferred option, definitely single use. That's very easy to use. What are your thoughts on then the data handling? Have you also worked on some models or some more sophisticated PAT solutions? Or would that be something that you're doing with your clients?
We would probably try to work with what our partners have. We have a whole group at CSEM focused on, it's called the AI for Life Sciences group, and they're specialized in getting these different data streams coming in and then helping integrate everything to make a decision. So we have the capabilities in-house to help do that, but ideally we would also be working with the partner to see what they already have, what their data management systems are.
Can you explain a bit more how you partner up with different organizations? What is your mission at CSEM and how someone maybe who's tuning in could potentially work with you and develop technologies?
We have several different collaboration models. The simplest is just kind of contract R&D, an industrial collaboration where we perform R&D as a service. The partner comes with a list of requirements. We brainstorm with them. We come up with a solution. Usually we start, if it's a new partner, we start with a small feasibility concept study, and then we go more into the development project. That's a lot like how traditional engineering companies work. What really sets us apart is that we can also access public funding like universities. Because we're a nonprofit in Switzerland under Article 15, we can access InnoSwiss, Bridge. We also apply to a lot of EU projects, also Swiss National Science Foundation projects as well. By also accessing public funding, it allows us and our partners to also de-risk the project significantly. For example, with an InnoSwiss project, you can get almost 50% of the cost of the project paid for by InnoSwiss. So now you're doubling your money. And what I've heard is that often we have more flexible IP conditions, some universities, and I think that also helps. In terms of technology transfer, we also often spin out our own startups. So we have internal funding from the government that we can use to develop core technologies. And depending on the best way to get this technology to the market, either it's through licensing or sometimes it's spinning it out. And so that's another way. If we spin out a startup that provides a technology that's of value to these partners, that's another way that they can also profit.
And what are your core competences?
Our main core competences, I would say, are customized consumables. So these types of fluidic lids, customized pipette tips, inserts for well plates, precision liquid handling, biomonitoring, and sample handling. So I can give some examples. In consumables, we talked about these fluidic lids. One of our most recent projects is a collaboration with EPFL in the FINV, and it's for an organoid dissociation tip. So if people are working with tissue samples or organoids or spheroids and you want to dissociate them into kind of single cell suspensions, this tip, it's compatible, designed for liquid handlers and pipettes, but you think of it kind of like a blender. You have a mechanical mechanism inside that helps to dissociate it, still in the presence of enzymes. But instead of waiting 20 to 40 minutes for your dissociation with an enzyme and pipetting up and down a million times, you pipette up with your enzyme, and in about 5 minutes, it's dissociated. We get slightly better dissociation into a single cell suspension, and we get much better cell viability because the time is decreased. So that's another example of kind of this customized labware. In liquid handling, we've developed a number of technologies that are now sold by companies like Tekon, Hamilton, these big liquid handling companies, Integra. In sample handling, that's one of our most recent startups, is an organoid handling and sorting startup called Visienko. And then biomonitoring, we've developed solutions, both optical with near infrared, mid infrared, also electrochemical. We developed a technology for CCIT, which is now, I think, part of Mettler Toledo. So I would say those four things are our core competences in the tools for life sciences. CSEM in general is much more broad, but I'll keep it focused on the life sciences.
And when should the company consider working with you to develop a new technology, and when would it not make sense?
So we are a early stage R&D factory, an idea and innovation factory. We don't do large scale manufacturing. We don't bring devices to market. We don't sell products. So we develop the initial concepts and bring it to a mature prototype level. It's around TRL 6, 7. We can then do some small series manufacturing depending on what the manufacturing is. We have a very powerful clean room at our headquarters in Neuchâtel. And so we can do small series manufacturing, producing tens, hundreds of wafers per year for customers. But beyond that, we then help transfer them to a foundry. If it's devices, we'll develop the first couple prototypes, but then we help transfer the partner to a contract manufacturing organization where then they will manufacture more of the devices on demand, also deal with regulatory CE marks, things like that. We do have ISO 13485 in our medtech division, so we can do that. But the manufacturing and scale up is, we usually do the transfer to another company for that.
For biotech startups or established pharma companies interested in collaborating with the CSEM, what partnership models have proven most successful, and what areas of bioprocessing innovation are you currently prioritizing?
The most successful collaboration models are definitely the direct partnership, but we're also very successful with InnoSwiss projects. So I think if you look at our statistics since 2018, we have a 70% success rate in proposal acceptance. And if the proposal is rejected the first time, we have an even higher success rate with resubmissions once you get the feedback from the reviewer. I think in recent memory, I only know of maybe one or two that were unsuccessful after resubmission. And then in terms of areas of bioprocessing innovation that we're currently prioritizing, I think the most exciting areas we've seen is definitely this microbioreactor topic, but then also organoid technologies. A lot of biotech pharma companies are exploring organoids in the drug development process, and there's a lot of gaps in the process. So, for example, this organoid sorting and handling technology, the dissociation, culturing, these microbioreactors could even be used for long-term organoid culture, because in a past collaboration we had with IOB, the Institute for Clinical and Molecular Ophthalmology in Basel, they work with retinal organoids, and they take 30 weeks to mature. And so if you could develop a medium exchange system, a lid, that just these organoids now sit in the incubator for 30 weeks and you don't have to open them, you don't risk contaminating them, that's a big value add.
Fantastic. This has been great, Tom. I really enjoyed this conversation. From all what we have covered today, what is the most important takeaway from our conversation?
I would say there's a lot of room still for innovation in this small scale microbioreactor field. There's a lot of companies that are active there as well, but I still think there's a lot of room for innovation still. Probably the two largest are in the liquid handling and in miniaturizing the biomonitoring strategies so that they fit. And once you can bring together liquid handling and miniaturized biomonitoring, I think you can have a really powerful technology.
Tell us where people can get a hold of you and potentially consider a collaboration with you.
The easiest is just to email me. It's thomas.valentin@csem.ch. And then also on LinkedIn is another easy way.
Excellent. Smart Biotech Scientist, I will leave all the infos in the show note. Please reach out to Tom. If this is something you consider collaborating, reach out. But also if you want to know more about perfusion technology, reach out to Tom. And thank you once again, Tom, for being on the show and sharing all these amazing insights.
Thanks very much. It was a pleasure.
That concludes our deep dive into small-scale perfusion cell culture with Tom Valentin. As Tom highlighted, the future of biologics development isn't just about the hardware, it's about intelligent data integration and predictive modeling that transform how we approach scale-up decisions. Are you struggling to implement these advanced process development equipment and strategies in your own development workflow? Book a free call with me to discover how you can fast-track your biotech development without the costly delays. I'll help you build a practical roadmap for bringing these continuous processing innovations into your specific workflow. Just visit BrumanConsulting.com/call to schedule your free call. And don't forget to subscribe on the platform you found us on and leave us a review if this episode helped simplify your bioprocessing journey. Until next time, remember, science may give you headaches, but biologic development should not. This is David Brulman signing off. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.